Okay, let's get started. Good afternoon, everybody. Thank you for coming. It is my great pleasure to welcome our very own Dr. Priscilla Hsue as today's CTSI CV-themed distinguished lecturer. As many of you know, Dr. Hsue was recruited recently from San Francisco to be the new chief of cardiology here at UCLA Health, and she is a professor of medicine and holds the Kawada Endowed Professorship in Cardiovascular Medicine at UCLA. By way of training, Dr. Hsue completed her bachelor's at Carnegie Mellon and moved to the University of California, San Francisco, where she spent the majority of her career. She completed her MD there, her residency in internal medicine, and subsequently a clinical cardiology fellowship, and then stayed on there as assistant professor and faculty. She quickly rose through the ranks and held the Maurice Eliasser Jr. Distinguished Professorship at UCSF, and was also appointed as Chief of Cardiology at the UCSF Zuckerberg San Francisco General Hospital, which is one of the busiest cardiology clinical hospitals in the entire Bay Area. Dr. Hsue's research and clinical interests, as many of you know, lie in the field of HIV, cardiovascular disease, and immune modulation, which she's going to talk about today. Her research interests intersect with acceleration of atherosclerosis and HIV, as well as HIV cardiomyopathy, and how this can teach us about other viral diseases, including affection of the heart in COVID-19. She's the PI of multiple cutting-edge, multi-center cutting-edge clinical trials, including ascertaining the role of low-dose methotrexate in HIV heart disease, and recently IL-1 beta inhibition with a monoclonal antibody, as well in attenuating HIV-related heart disease. She's received many, many honors throughout her career, too many for me to enumerate today. But for those of you who recently attended the American Society of Clinical Investigation meeting at Chicago last week, it's really an honor for all of us and for the institution that she was elected as president of the American Society of Clinical Investigation, an honor very, very few women have had in more than 100 years that the ASCI has existed in this country. So it's a real special that she was appointed. She is really somebody very special in the fact that she has real excellence in all three domains, what was called, nobody really uses this these days, but a triple threat in medicine, where threat is supposed to be a positive connotation, where you have excellence in research, teaching, as well as clinical care. So please join me in welcoming Dr. Hsue to the UCLA cardiovascular research community.
So just really want to thank Dr. Deb for that too kind introduction. And I've had like 10 cups of coffee, so I apologize if I'm going too fast, but I'm super honored to be here. Thank you so much. These are my disclosures. So I want to tell my story. So when I was in college, the HIV epidemic came out. I said, I want to go to medical school to UCSF. That's exactly what I did. I did a year of research with Warner Green at the Howard Hughes, and then pivoted into cardiology. And at that time, there were lots of young men coming in with heart attacks. And that made me wonder, is it the medication? Is it the disease? What is it? And it led to me starting in research, then looking at mechanisms, doing clinical trials, and I'll end by doing some of the future studies. And I'm wondering, who in this room is still training? Anyone? So for those who are still training, I just want to say, I've failed multiple times. And what I'm presenting here is kind of the nice version of what has happened. But, I want to say that it's a career in academics and research. It's a marathon, not a sprint. And you have to have that joy and love of what you do. And I super have always loved the research that I'm doing. So let's talk a little bit about HIV. This is data from UNAIDS, 88 million infected, about 40 million living, 42 million have died. In the United States, it's around 1 million living with HIV. And of course, this disproportionately affects individuals living in the South and from different ethnicities. It's crazy to think we've had 40 years of living with HIV. And so what are the recommendations for treatment? You should start treatment as soon as possible. You should start with integrase inhibitors as shown here. And there's new kind of targets that we should know the diagnosis of 95% of individuals for HIV, and they should start treatment and they should be controlled on medications. Those are the goals for the next 5 years. Where are we? So we're close, but not there yet. What about cardiovascular disease? So the first case was described in 1990, and let's flash forward 28 years later. This is a meta-analysis showing that individuals who have HIV are twice as likely to develop cardiovascular disease. The burden of this disease has actually tripled in the past 2 decades. And so that's shown here with this meta-analysis. So I have many take-home points. You know, the HIV population is aging. Inflammation and immune activation play a key role in this disease process. As cardiologists, HIV is a fantastic model of chronic inflammation. This can help us identify therapies for athero in general. And in turn, you know, for my HIV colleagues, ironically, understanding some of these inflammatory pathways can impact HIV cure. I'll talk a little bit about our studies of new viruses like COVID-19, our biologic and gene studies, and some of the future things we're thinking about doing. Let's talk a little bit about the description of the problem. So when I started as a fellow, I was really building that infrastructure to do research, training in advanced imaging, setting up mentors to do this. So what I did is I built this HIV cohort and used imaging that was non-invasive to characterize risk. And this was kind of establishing the center of excellence in vascular research with my mentor, Peter Gantz. And so this was doing things like endothelial function, arterial stiffness, echoes, and more recently CPET, you know, not only for our research investigations, but across UCSF, supported many different investigators and trainees. And so we know it takes a lot of time as clinicians to be exposed to something to develop clinical events like MI. So the first studies done of HIV were really using imaging like carotid IMT, coronary calcium, and all of these studies from my group and others showed that persons with HIV had accelerated and advanced atherosclerosis, including non-calcified plaque. And so shortly after that time, the study showed, okay, they had high events too. And this is a study from the Veterans Association showing individuals who have HIV have a 50% increased risk for acute MI. It is similar to that of potent traditional risk factors like hypertension and diabetes, and it's also common in young individuals as well. So the first part of the work in my team has really kind of established that field of cardiovascular disease and HIV. And this is whether it's atherosclerosis, HFpEF, sudden cardiac death, pulmonary hypertension, stroke, and you know, really bringing attention to this clinical issue. And so I think what's great as I've gotten older, the people that I've mentored are doing studies in HIV as well. And these are 2 individuals, Rushi Parikh, who's here at UCLA. I think what's great about this is these are interventional cardiologists that are now doing studies in HIV. And I think that's fantastic. They worked with one of my mentees at UCSF to further their studies. And so, you know, I think the cool thing on this slide, I got 3 interventional cardiologists to do work and research on HIV, and that is kind of something. And so I'm very proud of all of them. Now, the population with HIV is aging, and these are projections from this Dutch cohort showing if you fast forward to the next 5 years, that 12% of persons with HIV will be aged 70 and older, and over 70% will be aged 50 and older. And in fact, in the context of this, our group set up clinics looking at HIV with cardiovascular disease in San Francisco. These clinics have evolved with time. And so, you know, now something called Golden Compass. It's really a multidisciplinary clinic, you know, including HIV dermatology, neurology, geriatrics. And what's great is these clinics have really led to similar efforts across the world. So we're really happy about that. And so, of course, as the types of therapy used to treat HIV changes, so are the cardiovascular complications. You can think about before there was HIV medication, you saw things like dilated cardiomyopathy and pericardial effusion. Then in the area of protease inhibitors, we saw athero, stroke, and peripheral arterial disease. now with contemporary regimens, which are integrase inhibitors, seeing a lot of heart failure and arrhythmic complications. And of course, when you fast forward to the future, there'll be new medications, new side effects, but hopefully there'll also be curative strategies as well. So now I'd like to go to some of our investigations of mechanisms of this. And so, of course, traditional risk factors like smoking are very important and contribute to this. And this is a study showing that individuals with HIV lose more life years to smoking than they do to HIV infection. So that is important to know. There's also a role of the HIV medication itself. We know that protease inhibitors are associated with cardiovascular risk, probably a little bit independently of the dyslipidemia. Atazanavir may be protective because of high bilirubin. NRTIs, including abacavir, are associated with increased cardiovascular risk. And so the guidelines suggest that individuals who are at risk should likely be switched off these agents. And then finally, integrase inhibitors, which are first of line, are associated with lots of cardiometabolic issues like weight gain. And so the jury's still out on whether those agents increase cardiovascular risk. And then, so there is clearly risk that's associated with the degree of immunodeficiency in HIV. And this was early work done by Jen Ho, who is one of the first fellows I ever worked with and is now a very established investigator in HFpEF. And so our studies show that having a low CD4 count or advanced immunodeficiency is independently associated with cardiovascular risk. And this led to a whole series of studies showing that in the setting of effectively treated HIV infection, there's chronic inflammation that persists, and that strongly is predictive of clinical events. And interestingly, our team really established that key link, and it has led to looking at focus of inflammation as a cure for HIV. And I'm going to circle back to that at the end of my talk. And so I want to highlight some more recent studies that have broadened our understanding of the disease process, including imaging, looking at novel cohorts and specimens, and new biomarkers. So I want to first turn to imaging as a way to assess preclinical disease and evaluate pathogenesis. And so our group has looked at FDG-PET. And so this is a metabolically active tracer that's taken up by active tissues, inflammatory cells, and macrophages in the plaque. You can see an individual here with low uptake and on the bottom, high uptake. And so using this, we use this method to look at both arterial and hematopoietic activity in the setting of HIV. And here you don't have to be a radiologist to see that this is an uninfected individual on the top. And on the bottom, it's a person with HIV, and you can see how those lymph nodes just really light up. So there was a strong association between degree of HIV disease control and lymph node activity using this imaging. And then we looked at a way to look at HIV persistence, and using special assays, you can measure detection up to 1 copy per tube. And this is far more precise than what we do clinically. And so doing that, we saw that individuals who had elite control, so they had a strong association of this integrated DNA with lymph node inflammation, suggesting that these are important reservoirs for HIV disease activity. And we found that this was strongly associated with these degrees of viral control. So this study was really one of the first imaging studies to look at imaging as an assessment of the HIV reservoir. And building upon that, I think, my colleagues can be like, well, Priscilla, that was very nonspecific. What about an HIV specific tracer? And so this was developed with my colleagues at UCSF. This is radio-labeled zirconium that is a tracer developed to the HIV envelope protein, taken preclinical, then in animal studies, collaborating with my colleagues at UC Davis. This is zirconium VRC01. Here we have studied individuals first in human as well. And so using this, we coupled this with our ability to access tissue at UCSF. So we have a very active gut biopsy program, studied over 1,700 individuals. We're doing the same thing with lymph node tissue as well as bone marrow biopsy, and it's been very, very successful. And so kind of coupling that imaging to the tissue, we are doing kind of the first in human evaluation of this special HIV specific tracer, as shown here, with our colleagues at Davis. can see here a remake, a person with HIV that just lights up. And then importantly, coupling that imaging to assessments of the reservoir from gut tissue. And you can see here, this is very preliminary, but this is a nice association between the degree of viral persistence in gut tissue and our imaging using this PET MR. So more on this hopefully in the future. So with my clinician hat on, sudden cardiac death is a dreaded complication. And my colleague at UCSF was looking at this in the context of San Francisco. And he noticed that the first several individuals who died suddenly had HIV infection. And this led to this first study that was published now, like, a while ago, showing that, from this time period, 2001 to 2009, many individuals with HIV died of AIDS, as shown here in blue. But what was surprising is that persons with HIV had a high rate of sudden cardiac death, as shown here in red, and it was more than 4 times the general population. And it really made us wonder, like, why is that the case? And so we had a follow-up study to that, and it was really like 10 years of very painful work, looking at the reasons for sudden cardiac death for persons with HIV. And this is a very intensive study in which all individuals who had the WHO definition of sudden cardiac death were enrolled into the study. Very comprehensive emergency medicine review, toxicology review, medications. And then what was amazing, we had access to their tissues. So they had autopsy. And I want to point out, of the 109 individuals with HIV we studied, we got tissues on 108. And so that is really amazing. So this is, what were the causes of sudden cardiac death here? And so what was surprising, occult drug overdose caused a lot of these. And that's something that a defibrillator is not going to cure. But we also found other cardiovascular causes of both sudden cardiac death and then adjudicated sudden arrhythmic death, which included atherosclerosis, cardiomyopathy, as shown here. So I hate this figure because it's too busy, but long story short, persons with HIV had significantly higher rates of sudden cardiac death and autopsy adjudicated sudden arrhythmic death. These were findings driven by white men in our population. But I think the most important part of this is we had access to heart tissue, and this is staining of heart tissue and analysis. We know that the trichome staining here, fibrosis shown as blue. And again, long story short, all persons with HIV had higher levels of myocardial fibrosis. This seemed to underline the arrhythmic, sudden arrhythmic death. It was driven by interstitial fibrosis and not replacement fibrosis. And replacement is what happens in the setting of athero. So that was very interesting. This did not make the paper, but I thought was super interesting. And so we had nanostring analysis to look at gene expression. And in the study, we found that markers of macrophage activation were strongly associated with levels of myocardial fibrosis, suggesting that that chronic immune activation in the setting of HIV drives perhaps some of the things that we were finding. So I want to also point out that heart failure is common in the setting of HIV, both HFrEF and HFpEF. There have been some studies to suggest that HIV medication, like protease inhibitors, is associated with that risk of heart failure. With that in mind, we partnered with the Heart Failure Network to do studies of cardiac MRI to look at fibrosis and to look at some gut metabolites like TMAO. And TMAO plays a key role in HIV disease pathogenesis because HIV infects the CD4 cells of the gut, leading to microbial translocation and chronic inflammation. So this study showed that indices of TMAO were associated with myocardial fibrosis in the setting of HIV. These are 2 fellows who did the work who are now heart failure specialists at University of Pennsylvania and also OHSU. So this led us, again, try to do a deeper dive into this. So we made use of novel specimens to get access to novel tissues. So we partnered with the Last Gift study, which is a cohort at UCSD. It's persons who are living with HIV who have pre-donated their bodies to science, and they get autopsy within several hours after death. And so working with my colleagues there. And then also UCSF, our team has kind of very painfully but extensively banked all individuals who are undergoing heart transplant and LVAD studies. We banked around 60 individuals. And so some initial data from the collection of our samples So first, it was that we were able to get high quality specimens from our autopsy bank tissues. Here's an example of a person with HIV and an uninfected control. Neil Chi at UCSD did this analysis. You can see, first of all, we got excellent yield of nuclei from the cardiomyocytes of our HIV-infected individuals. You can see here, this is preliminary data, but there's clearly some difference in populations of lymphocytes, NK cells, and perhaps fibroblasts. And this is a collaboration that I have with Neil Chi, who's going to, look at disease in a dish models using our patient samples to model the impact of HIV medication, as well as inflammation and immune activation. And so this, again, hopefully we'll have more work to talk about in the future. So with this in mind, how do we predict individuals who are going to have problems in cardiovascular disease in the setting of HIV? I think it's not surprising that the risk calculators that we use in the general population do a very poor job and underestimate risk. And this is probably because they only focus on known pathways. And so with that in mind, we looked at a proteomics approach. And so what's good about that? So proteins, unlike genes, can change with the environment. They can tell us somewhat of the biology of the disease process. And in the general population, this approach has been shown to improve risk calculators. So we have partnered with my colleagues to use a precision medicine approach to develop risk prediction in HIV. This is using the SomaLogic platform, and this was done in the veterans cohort. So it's 1,500 individuals with HIV, around 850 without, and we had clinical events in this cohort. So these are some of our initial findings of proteins that are associated with mortality in HIV. And I know these plots are really, really hard to read because everything's on top of each other. Blue color is individuals who the proteins might be protective, and the red is proteins that are associated with mortality. But a better way to look at this is in a table form. And so these are some of the top 10 proteins associated with mortality in the setting of HIV. You can look at the biologic functions, and the proteins in yellow are the proteins that may be protective. And what's interesting is, a subset of these have known therapeutic targets. So of course, we modeled this to see, do the proteins retain importance after important clinical features? And they do. They also maintain importance looking at inflammatory coagulation biomarkers. And these are Kaplan-Meier curves showing that there is good prediction for mortality in both the training and testing set. So this was one of the first studies to use large-scale proteomics to look at clinical events in HIV. And Satay Shakil, who's a faculty member who came with me from UCSF to UCLA, has looked at proteomic signatures associated with heart failure. And here you can see there are novel proteins in the setting of HIV. Some of these have druggable targets as well. She presented this at ESC last year. So now I'd like to go on to some of the trials that my group has done. And so this is an old study, but lots of things come together to cause chronic inflammation in the setting of HIV. So HIV medication controls disease, but it doesn't cure it. So there's ongoing replication, there's toxicity from HIV medication, co-infection with viruses, microbial translocation, and then also immune abnormalities. All of these factors come together to cause chronic inflammation and comorbidities, including cardiovascular disease. There's been a whole ton of work showing that inflammatory and coagulation markers in many different cohorts and studies are strongly predictive of clinical events as well as mortality. And that is really striking given the variability of these assays and the differences in these cohorts. And in fact, some studies say that, well, if we could reduce things like IL-6 and D-dimer, this could translate to a significant reduction in mortality and help, with clinical events. So how are we going to get there? Should we look at things like statins? Those are, cardiologists love to talk about statins. So these are the cardiology guidelines for statin therapy, and these are before a big study. And you can see if you have this calculated risk that's between 5 to 7.5, you could say, well, your person with HIV, that could be a risk enhancer, and that would favor starting a moderate to high intensity statin. You could also think about imaging individuals to look at coronary calcium. So last year, there was a large study, it was very expensive, to look at pitavastatin in HIV infection. It It was stopped early because there was such a significant improvement in cardiovascular events as well as death, as shown here. And so I know there's probably very few clinicians in the audience, but if you are, does this study mean everyone with HIV infection should be on a statin? So yes, no, maybe. So I have my personal answer, and I think not everyone, but probably increased use among individuals who are between age 40 and 75. This will probably depend on your calculated risk. Now, what do I mean by that? These are the guidelines from societies about when to treat using an agent, when to consider it and not consider it. And it's based on this thing called number needed to treat. And so here, the overall number need to treat in this trial was around 105. And you can see that falls in the, you could maybe think about it, but one size didn't fit all here. So if you had a low calculated risk, probably less benefit. If you had a high calculated risk for cardiovascular disease, good benefit. So using those guidelines, if you're over the age of 40 and have a risk greater than 5%, you should probably be on a statin. And I do agree with that. New guidelines came out last year after that study, basically saying that same thing. And these are the guidelines in the general population. I think what is confusing is a low calculated risk in HIV doesn't mean that a statin isn't good at all. And that is, we know that from imaging studies, even if you have a low calculated risk, you might have atherosclerosis. And this was shown using carotid IMT as well as CTA imaging. So I think for some individuals, if you're on the fence, you could think about imaging them to consider statin use. So now I'd like to go to, we know that in the general population, everyone who has cardiovascular disease should be on high intensity statin. Some will benefit from residual lowering of cholesterol, and some will benefit from residual lowering of inflammation. I want to think about HIV in this context, looking first at cholesterol. So in the cholesterol world, we know we have PCSK9 inhibitors that are injectable agents that significantly reduce LDL cholesterol on top of a statin. And in the setting of HIV, individuals have higher levels of PCSK9. This mirrors levels of inflammation. And so thinking about that, I want to think about, and I think this actually even applies outside of HIV. Will there be a benefit to having a lower LDL cholesterol and doing this earlier? And, will this be beneficial in HIV? Of course, in the general population, lots of studies to show this using PCSK9 inhibitors. Fourier-Olay, which showed that starting PCSK9 earlier was associated with reduction in mortality. And clearly the benefit of statins in HIV appears to be driven by LDL lowering. And so will this be beneficial in our, the HIV infected population? So to try to investigate this, our group is doing a study of PCSK9 inhibition in HIV infection to look at the impact on arterial inflammation as well as non-calcified plaque. We are finally almost done with this, and so I hope to have some follow-up from this. We're also looking at different lipid-lowering agents, bempedoic acid, ATP citrase, lyase inhibitor. This has been shown to reduce events in the general population, and at both UCSF and UCLA, we are doing a clinical trial to study this in HIV. So I want to turn a little bit to inflammation in cardiovascular disease. There's 40 years of work, and now on the right-hand side, we finally have clinical trials. We're very early to try to understand the role of inflammation in HIV, and to date, there are no clinical endpoint studies looking at immunotherapy in the setting of HIV infection. So what do we know from the general population? We know that individuals who can lower both their LDL cholesterol and inflammatory markers, those individuals do the best clinically. And in fact, for a lot of individuals, you could consider additional cholesterol lowering and anti-inflammatory agents. So I think if you're a pessimist, you could say, well, that's great, Priscilla, those studies are old, not using state-of-the-art agents. So this roundtable looked at, what is residual inflammation using state-of-the-art lipid lowering? This is using PCSK9. You can see here in red and in orange that residual inflammation is still over 40% using state-of-the-art lipid lowering. And here, if you look at, reduction in LDL cholesterol on the x-axis and relative risk reduction on the y-axis, these are all our lipid lowering studies We can now put the HIV study here, but there's this group of orange studies, here on the left. They don't touch cholesterol at all, but they all significantly reduce cardiovascular risk. And so, this has led to Paul Ridker to say that combined use of lipid-lowering and anti-inflammatory agents would become standard of care for athero in the future. And so inflammation HIV doesn't just impact cardiovascular disease, it impacts cancer, kidney disease, mortality, premature aging. And so will then statins lower inflammation HIV? And so there have been a ton of studies to look at this now, including a larger study at the very bottom of 900 individuals. Statins seem to lower cholesterol, but they don't seem to impact the inflammatory pathways that we think are important in the setting of HIV infection. And this follow-up study showed that, again, pitavastatin lowered cholesterol in HIV, but it didn't touch those inflammatory and coagulation markers. So now I'm going to talk about some different studies of immunotherapies and anti-inflammatory agents. And so some will be broad, so looking at low-dose methotrexate. Some will be laser focused at innate immunity, like canakinumab, looking at IL-1 beta. And then finally, I'll circle back to colchicine. So I know there's going to be a lot of data, so I think seatbelts fastened. I'll try to deconstruct it. So our group looked at low-dose methotrexate in HIV infection, was to look at the impact on immune activation and inflammatory markers. This was the first study of immunosuppression in HIV. So there was a significant reduction in CD8 T cells, which is actually a target for HIV cure. However, didn't touch inflammatory coagulation markers. And so we published on that. And after that, the study of low-dose methotrexate in the general population, or the CERT study, was published, showing no impact on inflammatory markers and no impact on clinical events. We've had some follow-up studies to show, methotrexate might impact T cell proliferation in the setting of HIV. So again, looking at the inflammatory cascade, we've talked a little bit about statins that impact very downstream. Going upstream, IL-6, things like tocilizumab. I want to focus a little bit on IL-1 beta. So this is impacted by cholesterol, turbulent flow, neutrophil, extracellular traps, and hypoxia. There's a monoclonal antibody to IL-1 beta called canakinumab. So if we move upstream in this pathway, can we have a more potent impact on inflammation? And so we did a small pilot study looking at IL-1 beta inhibition HIV infection here. And first we shot using traditional parameters, no impact of this approach on HIV disease parameters. There was no impact on T cell activation. A single dose of IL-1 beta inhibition does significantly reduce both CRP and IL-6. And there was a significant reduction in both arterial inflammation, as shown here, as well as hematopoietic activity. And what about in the general population? What happened with canakinumab? And so here, there was this, canakinumab was studied in the general population, significant reduction in inflammatory markers and clinical events, didn't touch cholesterol, and it had an interesting impact on cancer, which I'll circle back to later. But due to a myriad of complicated reasons, it did not move forward for regulatory approval. So finally, what about colchicine? So it's something that's been around for decades, not sexy, but it did work in cardiology, I think surprisingly. So in the setting of acute MI, it reduced events in chronic cardiovascular disease. It's the first anti-inflammatory agent that's FDA approved to reduce inflammation in cardiovascular disease. But then I think some recent studies saying that in certain populations, patients with stenting, maybe it doesn't work. It's been studied in a small study in HIV showing no impact on inflammatory markers. So I think just turning back broadly, these studies that I've talked about in HIV can impact the larger population with cardiovascular disease. And that is, we know from Peter Libby and many others that both innate immunity and adaptive immunity play a role in atherosclerosis. We know from these studies that IL-1 beta inhibition might impact innate immunity, while methotrexate may play a role in adaptive immunity. These are studies that we learn using HIV as a chronic model for inflammation. And then finally, I think, from our cardiology textbooks, you remember of athero in general, you have the monocytes interacting with the endothelium, occlusive thrombosis and events. Clearly, there's many different pathways looking at atherosclerosis and HIV, and this could inform us about the whole role of immunology in the cardiovascular system, which is a very hot field. I think for my HIV colleagues, we have to think about, will these therapies and the target inflammation provide any insight into HIV cure? And so this is a vicious cycle type thing. You have HIV infection causes fibrosis, poor HIV disease control, and it's a chicken or egg issue. So if you cure HIV, inflammation will go away. And so that's clearly a very tough bar. But if inflammation causes viral persistence, then anti-inflammatory approaches could accelerate HIV cure. So working with others, we looked at mTOR inhibition with sirolimus, showing that in the small study, there was a significant reduction in levels of the reservoir that persisted after cessation of the study drug. And then we're also able to link markers of viral persistence with atherosclerosis in this study, suggesting then that therapies that target the reservoir could have an impact on cardiovascular risk. But I think I have humility when I mess with the immune system here. And so I've tried to present it as a very nice diagram. You have your HIV reservoirs at the bottom, macrophage monocyte activity in the middle of this tree, inflammation, and the leaves are end organ disease. And so, that's a nice theory, but I think in reality, it's likely much more complicated. So I want to turn to, canakinumab. I talked about it in the setting of cardiovascular disease. This is really confusing to me on immunology and how confusing this is, with my cardiology brain. And that is, there was a very interesting and unexpected finding in the CANTO study, and that is IL-1 beta inhibition had a significant reduction on lung cancer, fatal lung cancer, and all fatal cancers. And I really had to ask myself, why is that the case? And I thought about, well, if you reduce inflammation, can you reduce counter-regulatory immunosuppression, leading to a better control of cancer? And what do I mean about that? So I think what happens is inflammation's bad, okay, and it's inflammation here, it's your body stepping on the gas, and so it stimulates this potent and sustained immunosuppressive response, and that is immunoregulation, as shown here. That's your body stepping on the brakes. And that involves T regulatory cells, immune checkpoint receptors, and myeloid-derived suppressor cells. These immunosuppressive responses then can blunt responses to HIV and cancer. And so we looked at that in our canakinumab study in HIV. We found that levels of IL-1 beta in our samples were strongly associated with levels of the inducible reservoir. And there were a subset of individuals who got canakinumab who reduced their levels of the reservoir. This was studied by Jeff Tomulka at Emory, and he found that using bulk RNA-seq, that IL-1 beta led to changes in T cell function, and that was associated with lower levels of the HIV reservoir. And so, of course, these things that are good in HIV and cancer might be bad for the heart. And so that is, we know things like IL-10 is actually considered protective in the setting of atherosclerosis, same with TGF-beta. JAK-STAT inhibitors are great with reducing inflammation, but they're associated with thrombosis. And of course, the classic example of this is checkpoint inhibitors. We know they're fantastic in cancer, but they're associated with a myriad of cardiovascular complications. So now I'd like to turn to some of our studies in COVID-19. And so during the pandemic, we changed all of our expertise to look at SARS-CoV-2. Love this quote from Tony Fauci that, everything that we learn from other pathogens builds off what we learned from HIV. So our team looked at immunomodulation in acute COVID-19. This is us consenting a patient through the hospital door. I'll talk about some of these studies quickly. So we looked at colchicine in the setting of acute COVID in a very interesting outpatient study in which patients, it was all remote and virtual. They got couriered the colchicine to their door. So it was a super painful study to do, I will say. And it was before there was widespread testing available for COVID-19. So I think long story short, it did not seem to impact outcomes. But if you looked at patients who had, test confirmed COVID, this probably was beneficial. I think an important reminder, it's so important how you choose your endpoints in these clinical trials. So what about canakinumab in the setting of acute COVID-19? we looked at in this study. So again, long story short, no impact in the setting of hospitalized individuals. A lot of these individuals got other immunosuppressives like IL-6, and if you take away those others, it looks like canakinumab may have been beneficial. So pivoting from acute COVID, what about long COVID? We know that, after, having acute COVID, there were some reports of this in 2020, individuals that had persistent symptoms after acute COVID infection. This led to a large NIH effort. And so it's very common. It's about symptoms that are unexplained more than 3 months after acute infection, can cause severe debilitation, similar to other long viral syndromes like Ebola and other issues. And so, with this, the LINK collaborative was established, long-term impact of infection with novel coronavirus. I want to highlight Michael Peluso, who's the junior investigator at UCSF who put this together. You can see it's now 1,000 participants, incredibly well phenotyped. And we are trying to set up now LINK at UCLA with great pain, but I will someday be successful at that. So that study has provided tons of insight into the pathogenesis of long COVID. I think because of time, I'm going to just whoosh through those. And what's interesting is just like what I present with HIV, chronic inflammation is consistently identified in this disease process, as well as SARS-CoV-2 viral persistence. Now, at the beginning of my talk, I talked a little bit about our sampling for getting gut tissue, and we used that to look at some of the pathogenesis of long COVID. And so this is looking at gut biopsy specimens on pre-COVID controls. And here you can see now individuals with the detection of SARS-CoV-2 in their gut tissue post-COVID. And I want to point out some of these individuals are, almost up to 2 years post-COVID. And these are the immunohistochemistry. If you look at the immunostaining, they seem to co-localize with CD68, so monocyte or macrophage lineage, as well as some CD3, but a little less so. I think also when we did this in the gut biopsy, there was evidence of double-stranded DNA in the gut tissue in 3 out of the 4 individuals. And that suggests that active viral replication is going on now, even 2 years post-COVID. And so I think that was a super fascinating finding. My junior faculty, Matt Durstenfeld at UCSF, has looked at a lot of the cardiovascular sequelae of COVID using imaging and also arrhythmia monitoring. And again, long story short, found that pericardial effusions in the setting of long COVID were associated with symptoms, associated with inflammation. But he really focused on doing more detailed phenotyping using CPET, and he found that individuals with long COVID had decreased peak VO2, and this was found in other studies as well. And then digging more deeply into this, instead of deconditioning or cardiac issues, it was in specific chronotropic incompetence that was driving the decreased peak VO2 and symptoms. And this was also something that he's found in the setting of HIV infection. In both disease states, chronic inflammation seems to be associated with exercise capacity and also chronotropy. So I think similar to HIV, this chronic viral infection causes tissues invasion, chronic immune activation, inflammation, and these all come together to cause symptoms and long COVID. And each place on that pathway is targetable. And so this can be using antivirals, immunomodulation with IVIG and others, and anticoagulation. So our group is kind of honed in on looking at JAK-STAT inhibitors as a mechanism on this pathway for long COVID. And we did that because baricitinib, which is a JAK1/2 inhibitor, was FDA approved for acute COVID, and has also been shown to reduce mortality in hospitalized individuals. So working with my colleagues on Wes Ely and Vince Marconi at Vanderbilt and Emory, we are doing a 550-person clinical trial to look at the impact of JAK-STAT inhibition using baricitinib on long COVID. This will be a multi-center study, at 4 different sites, trying to bring it to UCLA, but again, trying to walk through the red tape on that. And so I think that will be one of the first studies looking at immunosuppression as a treatment for long COVID. And then finally here, I want to finish on some future studies. So when I was at UCSF, we'd spent all these years doing these really difficult studies to build that infrastructure to do first in human biologic and gene studies, the scientists, imaging, and we had done that in tissue banking. And so when I thought about UCLA, I think all the opportunities lie to build all of this and more here at UCLA, and that is super exciting. So I want to go back to HIV cure. So I talked about, all these millions of individuals infected. This is a little bit of an old slide. There's probably 7 individuals have been cured. You can do this by total eradication of the virus, or you can do this by, trying to control the virus and then, controlling it with medication. So with my, the last couple of years at UCSF, I wanted to do some first in human studies. And so I partnered with my HIV colleagues. And so we wanted to look at CAR T cells in the setting of HIV for cure. And so this is really genetically modifying the patient's own T cells with the duo CAR T cell approach to redirect them to bind to their HIV antigen and remove HIV from the reservoirs. So this was done in animal studies, in primate studies, and we tried to set up the infrastructure to do this at UCSF. And it turned out we had to pivot the trial to UC Davis because look at these requirements for monitoring for all CAR T cell studies at UCSF. In-house monitoring, your partner or the study participant cannot drive. And so we did that. There's been 4 persons treated. I think update the study, I think in 2025, a little bit of an FDA hold at this time, but I know the 4 first persons treated, there were no side effects, safely tolerated. I also got experience trying to set up the infrastructure to gene editing and in vivo gene editing for HIV cure. So, I'll try to make this fast. Basic science studies, doing unique guides, human T cells, mouse model to do this, non-human primates. The FDA cleared the first in human studies. So we set up the infrastructure to do this at UCSF, which took several years. And look at this protocol. It's very, very intensive. And so again, long story short, it took me 2 years to set up the contract. Eculizumab, which you need to have on hand in case your person has a toxicity from the AAV9, costs $100,000. No one wanted to pay for it. So the company got tired of us and they basically reached out to another private company in San Francisco, and they completed their studies here. So, they presented their studies like 2 years ago, has been well tolerated, and they have follow-up studies that are ongoing, and it's now on fast track. So I think I've learned a lot from this. I'm a non-oncologist trying to do some gene and cell-based therapy. I realized that all these protocols are built in the context of a fatal disease like cancer. So when we, as cardiologists and others, start to do this in non-fatal diseases, I do think we have to think about how do we develop and adapt that monitoring. And that's, of course, this is rapidly coming down the pipeline in cardiology, whether this is PCSK9, amyloid, and also now lipoprotein a. Okay, so in the last couple minutes, and I know I'm supposed to be finished because I see Lucy like, okay, quick on my new work that's ongoing. So we've looked at the role of clonal hematopoiesis of indeterminate potential in the setting of HIV. This has been described in the general population associated with cancer and cardiovascular disease and inflammation. I will just go through this quickly. So there are also some chip-related therapies that could be considered. These could be downstream, targeting IL-6, or more targeting chip more directly by NLRP3 inflammasome. So this has been described to be higher in HIV, and working with colleagues, Alex Bick, Pradeep Natarajan, and others, we are doing a large-scale study to look at the role of CHIP in HIV infection, looking at large cohorts, seeing the role of immune activation and the viral reservoir. Our group has also set up some new collaborations in setting of global studies. This is a study of sub-Saharan Africa, testing and treating for HIV infection, now looking at comorbidities, and would love to build global cardiology here at UCLA. Sateh Shakil is certainly doing a lot of this here at UCLA, looking at environmental exposures and novel mechanisms using imaging. And then one of the things that I also had wanted to do was set up research abroad. And this is some of the work that we're doing in the setting of Uganda, and it was a collaboration with Mulago Hospital and pulmonologists that have looked at TB infection. So we are looking at the sequelae of TB infection on cardiovascular disease. You could call it long TB. And this is a new grant that we got, and we're setting up the first CPET program in Uganda to be able to evaluate this. So I'll make this fast. Colette de Jong was very interested in polypill, and that is over-encapsulation of medication for heart failure and giving this to patients. She's now studied this, looking at stakeholders, and has also set up a clinical trial to do this. And this is, the design of her clinical trial, and this is leading to a PCORI study with Tommy Wang and others at UT Southwestern. I hope to do this here at UCLA, fingers crossed. And so in summary, I've talked a lot about immunomodulation and cardiovascular disease, the biology of inflammation, talked about different trials. There's tons of unanswered questions, and how do we bridge that? This is a nice statement that was put about by one of my mentees. And we need to train and support our junior investigators. Lots of things needed to move the field forward. And I'm gonna just whoosh past this. And then finally, I want to thank all of my team, my collaborators, and I'm super excited and honored to be here at UCLA. So thank you.
Philip, for the wide-ranging overview. So we'll have time for a few questions.
Yeah. Here you go.
Thank you. Very fantastic work. I want to ask you about sudden death in patients with HIV.
Yeah.
And you mentioned about the underlying mechanism could be interstitial fibrosis
Yep.
and its impact, and how that is associated with markers of macrophage activation. So from all the studies and the clinical trials and experimental evidence, any of these resulted in reduction in sudden cardiac death and underlying mechanism?
Fabulous question. I don't think, sudden cardiac death, the definition of it is a little bit imprecise at best. And so what we did was an autopsy adjudication to adjudicate what was thought to be sudden cardiac death and what was actually adjudicated arrhythmic death. I think the issue is in a lot of these trials, whether it's with statins and others, they don't have high rates of sudden cardiac death, and the adjudication, they might call it sudden cardiac death, but it could be drug overdose, right? Because I showed that as well. So we don't have a trial that's looking at the impact of any of these interventions on sudden cardiac death. But I think that's a great point. Yeah.
Priscilla, one question. So about the statin use in the HIV individuals. So we know that chronic statin use can actually lead to accelerated coronary calcification.
Yes. Yes.
And so I was thinking about that when you were presenting, and whether you had any data to suggest that, people with HIV and on statins actually have accelerated coronary artery calcification.
That's a fabulous question. So before statins, a lot of the imaging studies showed that persons with HIV have more non-calcified plaque. So this study that I mentioned, that was in the New England Journal, showed that, statins reduce clinical events, and they have an imaging substudy to show that it seems to reduce non-calcified plaque. But they don't have really longitudinal studies to look at whether statins may actually increase the rate of calcification, because that has been shown in the general population. So I think that's a great point, and will require these, longitudinal follow studies to ascertain that. Yeah.
Thank you. Really fantastic talk. So I'm interested in the collaboration you're working with Professor Chi at UC San Diego.
Yes.
Wondering if the plans for molecular phenotyping of these immunomarkers for HIV patients, and how are they contributing to the monitoring of the current patient disease status and guide therapeutics?
Yeah, that's a great question. So we he's doing analysis now on kind of patient tissues that we have, both from the UCSD cohort, which is kind of the last gift, as well as some tissues that we have collected also in persons with HIV who underwent heart transplant. So he's still doing it, and then he's also going to do, you know, the disease in a dish, the human pluripotent stem cell modeling as well. So he's still working on it. It looks like, I'm going to say that antiretroviral therapy may play a little bit less of a role, and it's probably more the chronic immune activation that is more important. But we are, you know, definitely looking at it using all types of techniques, including some, you know, looking at epigenetics and chromatin expression as well. So-
The UMAP you've shown,
Yes.
even though it's preliminary,
Very
fascinating.
It's interesting,
Mhm,
right?
mhm.
Yeah. So.
Priscilla, one more question, you know,
Yeah.
when you were presenting the data on the, on your New England paper with the Mason trichrome and
Yep.
fibrosis data. So, you know, what's interesting is that if you look at studies of sudden cardiac death in the UK and here, and which have been verified by autopsies, you see the same thing, that you see a lot of interstitial fibrosis. And that has gone on to generate this term, which is called primary myocardial fibrosis, as you know.
Right.
And I was just thinking whether you had any insights, either from that paper or from subsequent data, whether there was any correlation of, you know, a bunch of inflammatory markers you measured and interstitial fibrosis.
Yeah, that's a fabulous question. So I think, you know, some of the issues of that study is that, you know, the individuals might be down for a couple days, because this is a study of like looking at all persons who died suddenly in San Francisco. So this is not the one of UCSD. They had immediate autopsy. So as you can imagine, they don't get immediate blood draw. And so there's going to be a lot of, you know, we know this from study section, right? There's going to be a lot of like issues with measuring inflammatory markers in the serum that are circulating in that setting. And in fact, that's probably why the nanostring data, you know, because gene expression in a person who's been down for several days, right? So is it truly reflecting the biology, or is it a remnant of, right? Or is it a remnant of being down for several days? And so, yeah. But I will say because we had the trichrome staining and could compare it at least in the hearts of non-HIV. Like that was solid, you know, so for sure.
Okay, if no further questions, thank you.
Thank you. Thanks so much.
That was great.